In 1678, Antonie van Leeuwenhoek peered through hand-ground lenses and discovered a hidden world of little animalcules. Yet this discovery had zero impact on medical science at the time. The historical record shows these microscopic observations lay dormant for nearly 2 centuries before anyone recognized their importance to human health. The delay came down to a critical flaw in scientific logic. Identifying microscopic organisms near a sick patient does not prove that those organisms actually caused the illness. Physicians of the era had entirely different explanations. Many assumed these microbes were merely scavengers, harmless biological bystanders attracted to dying tissue, rather than the instigators of the rot. This confusion was compounded by the prevailing theory of spontaneous generation. The leading belief was that living microbes arose naturally from decomposing non-living matter, completely obscuring the true mechanisms of infection. Without a strict methodological framework to prove cause and effect, early microbiology stalled. The microscope remained a novelty for observing curiosities, rather than a practical weapon for treating disease. By the 19th century, medical theories were still dominated by concepts like miasma, the idea that diseases spread through toxic bad air. Physicians desperately needed physical proof of how infections actually moved from person to person. Progress required a rigorous, reproducible system of logic to link a single pathogen to a single disease. The primary architect of this new causal framework was a German scientist named Robert Koch. Koch set out to outline a universal, step-by-step test. His objective was to systematically strip away all biological variables until he could prove that one specific microscopic entity was the sole cause of an illness. Moving away from assumptions about correlations and into the strict proof of causation provided the bedrock that made modern clinical medicine possible. But testing a suspected pathogen requires following a core rule of the scientific method. You have to isolate your variable from all the surrounding biological noise. Early researchers relied on liquid broth cultures. These environments allowed dozens of different microbial species to mix and multiply chaotically together, making it impossible to single out the true culprit of an infection. Koch solved this bottleneck by introducing solid media for bacterial growth. Unlike a liquid where everything mixes, a solid surface forces individual microbes to remain fixed in one location as they multiply, eventually forming distinct visible colonies. To further differentiate these organisms under the lens, Koch also introduced new chemical staining techniques. These innovations allowed scientists to finally achieve a pure culture, a biological population consisting entirely of a single species of microorganism. Achieving this pure culture was the mandatory first step. You cannot definitively prove a specific microbe causes a disease until you can successfully separate it from the pack. With isolation possible, Koch published his definitive 4-step algorithm for proving biological causation, now known as Koch's postulates. Step 1. The organism must be consistently associated with the lesions of the disease in every case. Step 2. The researcher must isolate that organism from the diseased host and grow it in a pure culture. Step 3. When that isolated organism is inoculated into a healthy, susceptible laboratory animal, it must produce the exact same disease. And Step 4. The organism must be physically re-isolated from that newly infected experimental animal. Koch applied this exact sequence to the tubercle bacillus. He tracked the bacteria from a damaged lung, isolated it on a plate, infected a healthy animal, and retrieved the identical bacteria from the new infection. This closed-loop system offered incontrovertible, mathematical-style proof of biological causation, finally silencing the scientific skeptics of his era. But a scientific law faces a crisis when a deadly pathogen simply refuses to follow the algorithm. The bacteria responsible for leprosy, the leprobacillus, breaks the second rule. It causes catastrophic illness, but it cannot be grown in a pure culture outside of a living host. A second crisis emerged late in the 19th century with illnesses like tobacco mosaic disease and yellow fever. In these cases, researchers could find absolutely no bacteria at all. Scientists used porcelain bacterial filters designed to trap all known microbes. Yet the causative agents of these specific diseases slipped effortlessly through the microscopic pores. These ultra-microscopic entities were viruses. Because they were far too small to be seen with standard light, they completely bypassed the optical limits of Koch's era. It required an entirely new technological leap, the invention of the electron microscope in the 1930s, to finally visually confirm what the filtration experiment suggested. Koch's postulates laid the necessary logical foundation, yet the physical realities of the microbial world forced researchers to invent increasingly advanced tools to prove causation at ever smaller scales. Once causation is definitively proven, the logical next step is clinical, learning how to destroy the specific pathogen without killing the human host carrying it. German scientist Paul Ehrlich took up this challenge. Much of his early work involved applying chemical stains to cells and tissues to study their functions. Ehrlich made a pivotal observation in the lab. He noticed that certain chemical stains would selectively color specific bacteria while leaving the surrounding human tissue completely untouched. He reported the acid-fast nature of the tubercle bacillus, proving that different pathogens possess unique, identifiable chemical properties. Ehrlich connected his staining observations to the potential for therapy. If a compound selectively colors a specific microbe, a toxic chemical could be engineered to selectively destroy it. His side chain theory proposed that cells possess unique chemical receptors, allowing a drug to latch on to a specific pathogen without affecting the surrounding human tissue. Understanding biological specificity, isolating the chemical identity of the pathogen from the host, provided the crucial bridge between diagnosing a disease and actually curing it. Ehrlich's theory culminated in his pursuit of what he called the magic bullet. This marked the birth of chemotherapy in its original context, the use of standardized, targeted chemical compounds deployed to hunt down and destroy a specific biological invader. His efforts resulted in the discovery of salvarsan, an arsenical compound capable of destroying the spirocate that causes syphilis, while keeping the infected patient alive. The historical progression is clear. It began with Leeuwenhoek passively observing microscopic life, advanced to Koch building a rigid framework to prove causality, and arrived at Ehrlich engineering targeted destruction. This exact sequence still forms the operative blueprint used by modern pharmacologists and immunologists today. Demanding proof of causality turned a descriptive science into a predictive one. Modern pharmacology rests on this ability to isolate a cause and engineer its specific destruction.